Hi, everybody. This is Dr. Ed Park, and welcome to another podcast. This is podcast 37 on telomere measurements. People look at telomere measurements, and a lot of people on the call have had their telomeres measured by us or elsewhere. And it can be kind of shocking if the numbers are good or bad or anything. People are very literal, and they look at telomere measurements sort of like a parking meter, and that when it goes down, you expire. And that is nothing could be farther from the truth. It's more like an odometer, but an odometer also doesn't tell you when your car is going to die. Witness this gentleman who drove his Volvo for 2.4 million miles and counting. And this is really the kind of paradigm we need when we think about what we're trying to achieve here. We're trying to keep the odometer going by replacing all the parts, by keeping them factory new, well oiled, and that's really the goal here. It's not immortality or becoming anything divine, it's just taking care of the car. So what is a normal telomere measurement? Normally, when the fertilized egg is created from the sperm and egg, you start at 15,000, more or less, in humans. And it's literally more or less. In mice, it's somewhere in the 30 range, and that's because mice have been bred for long telomeres, and so they make a particularly poor aging model, except with the engineered mice that we'll discuss later. So think of it like a pinball machine. And since all the eggs are begat from one fertilized egg, they all inherit the 15,000. As the cells copy and differentiate, they lose telomeres, and on the average they lose 50 to 100 base pairs per division. So what does that mean? Because of the mechanics of copying DNA, you can't get to the very end and you lose 50 to 100 base pairs. A base pair would be an A plus a T on the other strand, or a G matched with a C. So DNA is, of course, like a ladder. So how can it be that you lose 50 to 100 base pairs per division, cells divide millions of times, and yet you still only lose 15 to 100 base pairs per year? That should be completely illogical. And the reason for that is because certain cells, called stem cells, are endowed with the ability to lengthen. So don't think of it as a literal measurement. It's as though you're walking up an escalator. So you are keeping up with the loss because the stem cells, which are the queen bees, are keeping up with the loss. If you look at the DNA here, which is all folded up and squiggled, this is a chromosome. In the middle is the centromere, which is the center body, which is used to manipulate the chromosome when it's divided. At the tippy tip is the end, which is folded in on itself just like a paperclip, literally. So here is what the ultrastructure looks like. It's folded in, and that's so it's like an aglet on a shoelace. You can't have the loose ends or they'll fray. So the plastic tip keeps it from fraying. Moving right along, the concept of replicative senescence is an important one, and that means that as the cells divide, they get older. As cells divide repeatedly, they're copying from the previous generation, and they lose quality. In photoreproduction, this is referred to as generation loss, which is shown graphically by repeatedly copying this image. Keep in mind, non-stem cells that don't have telomerase, that's okay. But when the stem cells start to have that problem, we have an issue. So here's a blue chromosome and a red chromosome. They've both gotten so short that the ends are exposed now. And the mechanism to watch the DNA doesn't know if that's a break. For all it knows, it got hit by radiation from the sun, it's chopped in half, and it needs to be reconnected. So the mechanism of repair puts the ends together, and you form this double chromosome, shown here. So when a telomere erodes to the tip, it will recombine into a double chromosome, much like these two trapeze artists. Now, if you think of these trapeze artists as crazy glued at the hands, and you pull them apart, they could come apart at the shoulders, at the hips, not necessarily at the hands. So that's what happens when the cell divides. The chromosomes all line up, and they get pulled apart. And if you have a double chromosome, they'll get torn. So here, the tip of chromosome 1, which is represented in yellow, has been combined to 4. The tip of 4, or the short arm, has been connected to 5. And this is the process which occurs at the end of every cell's life. And it's normal, and it's healthy, and it's fine. You can result in a loss of 7. See, 7 got distributed to 3 and 4 here. And you can get triple copies. 13 has 3 copies. And basically, the colors will tell you everything you need to know of where the chromosomes came from and where they went. And so that's what we're talking about in terms of replicative senescence. That's the ultimate result. But as I said, self-destruction is good. It's like the Mr. Phelps tape that would self-destruct. When the tape reaches the end and there is no protection, the chromosome will signal destruction of the cell. So this is reflected here. If you put cells in a dish and feed them, after about 50 divisions, meaning mom makes $2, the $2 make $2, etc., you'll start to die off after about 50 divisions. That's the Hayflick limit, named after Leonard Hayflick in the 70s. And the growth of the aneuploid fraction, which means the percentage of messed up chromosomes, like the ones we saw from this mishmash. This is actually from a patient's small cell cancer of the lung. That fraction goes up, which is obviously the cause of the demise of these cells. So let's look at differentiation. Zygote is the fertilized egg. That can become and does become every cell in the body. And as it differentiates, it loses potentiality. It becomes maybe 3 layers, then, yada, 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 till the very tip where some cells have no potential. And as we lose the potential to become all things to all organs, we lose the telomerase activity as well. The way that the self-destruction is mediated is that the chromosome has checkpoints in its life cycle. The checkpoint occurs, and so if there's a problem with the chromosome number, if we go back to this slide, where this is an actual division of a cell, this is one of the checkpoints where you're checking to see if all the little chromosomes are present and accounted for. And if the tension on these spindles is not correct, the cell will not divide. And furthermore, if there's abnormalities, the cell will signal its need to die. So end-to-end fusion is what we talked about before, non-homologous end-to-end joining, and that causes apoptosis. You can do it through the p53, which is shown here at master watchman of the DNA, and it will poke holes in the mitochondria, which are like little battery acid packs, and that will cause oxidative damage, and the thing will melt like the Wicked Witch of the West, having water thrown upon her. So that's what happens with the cells. They fry themselves from inside, but only as a response to the accumulated DNA damage, which triggers a p53. There's another pathway, the p21 and 16, I think, which is aging, and a lot of the organ failures because the cells are not able to kill themselves. They just hang around and cause trouble. Okay, that's pretty sciencey, but let's go back to analogy. So a stem cell is like a queen bee. A queen bee is living years, and the drones live months. So she has immortal telomerase activity, so to speak. So these are all clones of her, and we don't care that in 50 generations they will be dead because they are unimportant. And that's how we reconcile the paradox of losing 50 base pairs. Yeah, the drones lose 50 to 100 base pairs, and yeah, the drones will be dead in 50 cycles, but the queen, not so much. She has the telomerase activity to live forever. So what is this? This is a representation of what an organ would be. An organ is a collection of many queens in their niches. So if we think of this as the liver, let's pretend that there's only 9 kingdoms or queendoms in the liver. You're not going to get liver cancer everywhere at once. You're going to get it here in this right upper lobe. And then all the drones will have her genetic abnormality and her telomere length. So if you were to cut a cross-section through here, you would see that the average telomere length here is 8,000, 6,000, 4,000. And here, it might be some crazy number that would be normal, but you'd have extremely long and extremely short. And more importantly, they'd have the characteristics of cancer. So that's really what we're talking about. And so we understand that disease comes from a single cell, and so it really boils down to one little error on one chromosome can cause you to die, and that's pretty serious. Okay, so Nobel Prize. The Nobel Prize in Medicine in 2009 was awarded to Blackburn, Greider, and Shostak, and they found that telomerase was an enzyme present in all cells, in fact all plant, animal, and fungi cells, to protect the telomeres. And this is huge because this is how all stem cells maintain viability. So what is telomerase? Telomerase is this blob. And people ask me, well, why can't I just eat telomerase? Well, telomerase is not that kind of thing. It's an engine comprised of multiple protein molecules. So it's this purple, blue, yellow glob. It's a machine, and the machine's purpose is to print out new base pairs in the telomeres, 6 at a time. And the reason why you can't eat telomerase is because this is only assembled within a cell for use only in the chromosomes for that particular stem cell. So when you take TA65 or a telomerase activator, you're making more of this stuff, but the problem is the key. This molecule called discarin, that's the keyhole, and the key is this corkscrew end of this green molecule. The green molecule is TERC, T-E-R-C, telomerase RNA component. This is the key. So basically, non-stem cells, the key is too long. So it can't operate the keyhole, it can't free up the active site to print out more telomere. Stem cells, they have a special splicing which makes it shorter, fits the keyhole, the active site is open, and the engine can grind out more telomere length. So what we have here represented on the upper left is regular drone cell, green. 50 divisions, it'll die. Premature aging syndromes, such as progeria, it's premature aging. So we know if nature gives you only one copy, you're going to die of old age, cancer, Alzheimer's at the age of 12. So one copy of this good TERC or green key, you're going to live 12 years. 2 copies, 80 years. 3 copies or more, who knows? So that's kind of the explanation there. So I think that you can argue that there's only really one disease process, and at the heart of it is the replicated senescence that we all understand very well right now. So that the chromosomes ends shorten like the fuses on firecrackers, and when that happens, they get DNA damage from that double chromosome phenomenon, and then they fail to kill themselves. This third ring should be failed apoptosis, so they sit there and they don't play nicely with other cells, and they copy themselves into naughty drones. So what is that manifestation in organs? Well, in the brain, it would be Alzheimer's, and so the organ level failure, like let's say we had the liver here, we talked about it would be liver cancer or cirrhosis. In the pancreas, diabetes, gonads, menopause, wrinkles, cancer in stem cells generally, failed immunity, macular degeneration, etc. So it's really just one disease, I believe, that's going on. And that's what the literature is showing. And so we don't judge the health of our dogs based on their hair. And so what I'm trying to say here is you got a healthy Westchester Kennel doggy here with the long hair. You got a beautiful Dachshund with the short hair. And you got a sick doggy with the mange. And this is what we're talking about when we talk about telomeres. We don't care if your chromosomes are super long, although that's nice. They have longer time before they get to the ends. We don't care if it's super short, as long as they're all there and there's no bald spots. But the mange is what would roughly correspond to this: critically short telomere, recombination, breakage, and chromosomal abnormality. So another analogy would be tires. Yeah, we'd like to have great tread on our tires, but as we get older or drive, in this analogy, the tire wears out. But we don't really care if it's a nice, smoothly worn tire. If it's uneven, or if we get a hole, that's really where the rubber meets the road, so to speak. So this would be analogous to one single chromosome tip, and so that can cause havoc if the cell is already immortal queen bee stem cell. Here is healthy long chromosomes. See how they light up at the end from a real cell? And these aren't abnormal, but let's pretend that they are shortened abnormal red telomeres here. And then our small cell carcinoma. And keep in mind that cells can exist like this for a while if certain self-regulatory mechanisms are gone and the ability to announce their abnormality is thwarted. The immune system can't destroy them. And even though they're broken like this, these cells can still function. The genes are still in the wrong place, but they're still being transcribed. So as we get older, this stuff is accumulating all over. And good example would be myelodysplasia. Myelodysplasia looks like this. Eventually, you have to understand it's all the same disease in different organs. And so there's 15,000 articles in the literature saying, don't let your chromosomes get short. It causes all the diseases, and that's really we're due for a paradigm shift. So this is kind of cool, these helical candles, and they happen to be colored like this fluorescent study on the chromosomes. So melting chromosomes plus time equals disease. Okay, so Nobel 2009, Nobel 1946. This gentleman, Hermann Müller, worked in Woods Hole, and he irradiated fruit flies with ultraviolet rays. And what he found was that he irradiated them and caused them to mutate. So he got a bunch of these mutations in the fruit fly's DNA. And he concluded, because he never saw there was a naked end, he always saw these end bodies, which he called telomeres, so he named the telomere. He never found mutations with deletions or inversions at the ends. The ends are always capped, or in the case of the double chromosomes, they were recombined or spliced inappropriately. So he concluded that the terminal gene, or the telomere, must have a special function, that of sealing off the end of the chromosome, so to speak. Not so to speak, actually literally, he didn't know at that time, and that for some reason the chromosome cannot persist indefinitely without having its ends thus sealed. Smart guy. Here's another experiment from a very smart lady, Maria Blasco, I think 2007, and she made these mice. The problem with using mice is that they've been genetically inbred and they have particularly long chromosomes, and they die and they get cancer and the disease, but telomere shortening is not a prominent factor of them, so they make a particularly poor model for us to study. On the other hand, if you breed them to have a mutated RNA component, remember that's that green corkscrew key, then every generation will age, so it will inherit the telomere length of the previous generation. So what we have here is G3 is the third generation of telomerase negative mice. Every child is born older, and then the third generation is really quite susceptible to aging disease. So this is a better model, which was also developed at Woods Hole. And so what she found was when she spliced in with gene therapy or transgenic mutation, a fresh new copy of a key that works, and all of a sudden the stem cells could use their telomerase because they had one functioning copy. That's why it's plus and minus. So they were progeric, but still had the functioning telomerase anti-aging mechanism. So they would be like those 12-year-old progeric kids. What she found was when you introduced it, the signal-free ends dropped off to zero. The rings, all these weird mutations that come from splicing and erosion dropped off. The end-to-end fusions, which is what we talked about with the trapeze artists, 0 from 16. So there's something going on here, which Müller alluded to, which is if you have a functioning healthy mechanism, you shouldn't have these end-to-end fusions, and these the fragments were increased and the breaks were increased for reasons which elude me. But in this is a study where she was able to drill down to the individual telomere length level, and again she found that the mean telomere length increased when you introduced functioning telomerase activity, and the percentage of short telomeres decreased. And this is really the genesis of her efforts to measure shortest telomeres, which have come to fruition in the life length test, which is we'll talk about a bit later. This is the slide everybody should know, but it looks very daunting. Suffice it to say that TRF stands for terminal restriction fragment, and this is very good for measuring things like carrier genes for different sequences and for finding out if criminals are related to their blood. So all the studies in the literature in the last 20 years were on TRF, and this is a particularly poor method of study. It requires quite a number of cells, but look at the resolution. There's plus or minus 1,000 base pairs. That's a pretty imprecise test, even though this is the quote-unquote gold standard. The interassay tests, meaning you do one in Cleveland, I do one in Detroit, there could be up to 10% difference in what we're measuring on the same person, same sample. So that the gold standard is poor. Stella is single telomere analysis, and that uses only the XY 2 and 17 certain chromosomes. So you're only really looking at a sampling, not every single chromosome. And it's particularly poor if people have variants, and it also overestimates or underestimates depending on this variable region. Resolution is good, but it's like getting 5 pieces to a jigsaw puzzle instead of all 10 or something. Here's a representation of the telomere. So the blue is just the stuff that goes on and on, 2, 4, 6, 10, up to 20,000 base pairs. This variable region has some of the blue sequence, which is GGG-TTA, but there's also junk mixed in, and this is the X region, or the unknown region. And then below that, or closer to the middle of the telomere, or the centromere, is the subtelomeric region, which can run up to 3,000 base pairs. So some of these techniques, like QFISH, they can measure and include the X region, which will cause a overestimation of the functional telomere length by up to 2,000 base pairs on average. So because there is variation here, you might get a report from a certain lab, and it might not be what you need. So the commercially available test using quantitative PCR, even in its more reliable iteration, MMQPCR, is the SpectraCell, available domestically for several $100s. The problem with SpectraCell is that look at the correlation to the gold standard being not very good, but if you repeat it with these two tests, the correlation is only 60% or 80% with the higher resolution MMQPCR. So there's a huge amount of variance there. It's not a very good way to measure. The inter-assay variability, meaning your lab in Cleveland, mine in Detroit, is up to 3%. And then intra, meaning same person, same lab, same specimen, up to 5% with both methods. So there's a huge error there. So there's something wrong with the qPCR technology, in my opinion, which SpectraCell would not agree with. But part of it might have to do with the fact that PCR is good for amplifying really small traces of DNA, but there's an echo factor here because it's all repeating. So a better way to do it is QFISH, quantitative fluorescent in situ hybridization. So what does that fancy word mean? Well, in situ hybridization means you take apart the strands and you anneal a complementary strand here to the GGG TTA. Would be CCC AAT, I guess, running in the opposite direction. So it sticks it. It really labels it, and then it fluoresces. So it's fluorescent. It's in situ, right to the actual DNA, and it's hybridized. It's making a ladder, a double helix again. So this will paint anything that is the right sequence, and he quantitates it. The amount of light coming back is the amount of telomere, QFISH. And so let's look at the resolution here. It's plus or minus 0.3 kilobase pairs. So it's only 300 base pairs. So that's much better than the 1,000 on the gold standard. It correlates 90% with the gold standard, so that's really much better than qPCR, which is 60%. And the intra-user variability, it's 11%. It's pretty high. So what do we do to correct that? Well, we get more cells. We get a ton of cells. We do a high throughput flow FISH, and that takes the inter-assay coefficient of variation down to 1%. So you do yours in Detroit, I do mine in Cleveland, and we're going to be within 1.6% of each other. So this is a much better test, much better even than the gold standard. So this would be Lansdorp in Vancouver, the repeat diagnostic test, which we've been using since 2007, and which is highly reproducible and highly accurate for median telomere length. So again, the correlation, about 0.9. Very good. One that's not mentioned here, this is a paper published last year by Dr. Aubert, who is a principal in the lab of Dr. Lansdorp, and it really is a wonderful review article. Aubert spelled Aubert. Now, she doesn't mention the further modification that Glasgow's lab has made, which is high throughput flow fish with automated microscopy. So let's see what that looks like. That's the life length test that became available in January of this year. Oh, this is people ask, well, is this really reliable? Well, if you look at the high throughput flow fish that Aubert and Lansdorp do, this 45-degree beautiful line is the correlation between measuring one off view and measuring the other. It's the same. Pretty much the correlation is 0.99%. So the precision is quite good. And so this is represented here. If we have a standard known length telomere, we run it simultaneously with the patient sample. It's very, very reproducible. So these measurements are quite precise. So this is a picture of me, pretty heavy, very stressed out, high blood pressure, low testosterone, fatty liver, high cholesterol, prediabetes. And this is, the result of poor lifestyle choices, I would say. My son is 1 here, and so I'm only 32 years old, but I looked this way since residency 26. That was really quite difficult, staying up, 36 hours, working, stress, making sure people's kids don't die in childbirth. And so it's put me into this metabolic syndrome. And also, I got married at that time and was diagnosed with sleep apnea. My wife recognized that I wasn't sleeping well, and I would choke, awake several times an hour and snoring. And so at that time, sleep apnea was a relatively new diagnosis, but I did a sleep study, was diagnosed with that, got a machine, and for the next 18 years, I slept happily ever after, which I would attribute as the reason for my extremely long telomere length. In 2007, when I was the 19th person in the world to start the patent protocol on the TA65, I measured at about 9100 or 9200. And then they would repeat the test because they didn't believe it was so good, and the repeat would always be, I would always get 2 for 1. It would always be within 100 base pairs. So it is a very precise test. So in 2008, there were a lot of stresses in my life. It dropped to 8,400 base pairs. Now, would that have been 7,200? If I hadn't taken the TA, I don't know. But what I can say is I didn't panic because I had lost 20 pounds. I was feeling great, better than I ever did since early 20s. And so I had to go with what was real, which was my appearance, my function, my mind, all my cholesterol, testosterone, fatty liver. That all went away, or normalized. So I didn't panic, even though it would appear I was 7 years older by chromosome length. And there's other explanations we can go into, but I stayed the course. And most recently, at the beginning of this year, we did the life length test, and it was 9,800. So it's a moving target. Certainly if your spouse is diagnosed with cancer, you're up at night, stressed out, not sleeping. I guarantee you're not doing the repair you need to do while you're sleeping, and your telomeres are eroding, so they can erode quickly. And again, I don't want to be obsessed over the median length, although it is important, but I have to reiterate it. One single chromosome can kill you if it's in the right place and if it's not controlled properly. So 12 years later, I feel 20 years younger. I'm not sure what kind of swing that is. That's my son, who is now 13. And so still not exercising or dieting regularly or anything, but it's really difficult for me to put on weight. So that tells me that in the visceral fat, the omental fat, some of those queen bees were killed, never to return again, because I actually stopped taking in 2010 for about 9 months, and even in that time I couldn't gain weight, whereas the gentleman or the guy, the fat dude here, who just sniffed a Kit Kat bar would gain 5 pounds. So something changed with my metabolic syndrome to now I can, drive through and get fast food and it really won't affect me. So I have the fat stem cells of a younger person. And indeed that's borne out in the leukocyte telomere length, or the white blood cell telomere length. So this is an example of a life length test. Here's the median length, 9.8 thousand, which would be better than even around a teenager. If you really want to extrapolate, say you're born with 10,000, that would be a 3-year-old. What's different about the life length test is they have a second variable now, which is the percentage of short telomeres, defined here as less than 3,000 kilobase pairs. Now, once you get to 2,000, whether you're counting the X factor or not, or the X region, the cells really can't be sustained with less than 2,000 or 3,000 base pairs. They become exposed and cause damage. So my percentage of short telomeres in the white blood cells, or leukocytes, is 10%, which puts me again in the teenage range. We don't know what it was in 2007, but presumably it would have been higher. Here is the picture of my actual telomeres on my actual white blood cells. And again, here's a nice line showing that at the estimated age, this should be the estimated, this is the telomere length for my chronological age. It doesn't match the line. It's like a 20-year-old, even though I'm 45. So here's some pitfalls. I got this garbage in, garbage out slide. I think it's a computer something or other science. But this is very relevant because people are always calling me and saying, Doc, I only got so-and-so years to live and blah, 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 and should I take the TA, should I not take the TA, what if it got worse? And the problem is that people have a lot of false assumptions. So let's talk about these assumptions. Garbage in, garbage out is a saying, and some people would take it to mean, well, your result is only as good as your data. Well, it's only as good as your data, and it's only good as your model, too. And our model for understanding telomere length is completely non-existent. So let's look at why we have problems interpreting these telomere tests. One is a linearity assumption, which is that telomeres only shorten, and they shorten at a predictable rate. Totally false. One study by the Nobel winner showed that women who are stressed and taking care of disabled children, they age faster. And this stands to reason. It's what we know to be true, that when people are stressed, they don't sleep well, and then at night the chromosomes don't repair. Also, other variables may not be considered. We're taking the leukocyte telomere test because it's easy to draw a person's blood, but are we considering the variable that there might be poor correlation with other organs? If we're looking at a woman's risk of cervical cancer, we're not sampling the cervical cells. If we're looking at an alcoholic's esophagus or liver, we don't study those. So the presumption is that there is good tight correlation between the blood measurement, which is, on the face of it, false, I believe. Negligibility assumptions, like we think that certain things are not important when they might be. Our data might be bad, again, if we're using QFISH, and even with good data, our understanding of it might not be proper. So again, there's interlab variability, which is huge. We talked about that. Also, if you think about it, if you just had a bout of flu or a bacterial infection, that will shift your leukocyte telomere length too, because there's a sudden proliferation, a clonal expansion of the white cells. So you get a lot of fresh recruits, so that might change the way it looks. These things are totally unaccounted for. So you really can't take one result and say, the sky is falling, I'm going to die here. It's a trend, so we like to encourage multiple measurements. Now, having said that, the new method with the shortest telomeres, I do believe, will be quite an interesting usage of technology. So the last two slides, let's just step back and take a breath and let's talk about what the heck is going on here. So certainly if we stimulate telomerase activity, which TA65 definitely does, independently verified. Wouldn't that just protect the ends and prevent future damage? Yes. So why would you get an effect of reducing the shortest telomeres? Well, I like analogies, and the best analogy would be, let's say you got a broken motorcycle or a lawnmower, and there's a screw here you need to get rid of to kill that cell or to destroy that bad stem cell. If it's stripped here, how are you going to get rid of that? So I think something about telomerase activation allows the telomeres to be manipulatable, if that's a word, and someday people will look back maybe and say, oh, okay, this is what he was talking about. But when you look at the clues here, BRCA, which is the famous BRCA, Berkeley Research, or breast cancer, Susan Komen gene, what that really does, one of its functions, is to handle telomeres. It localizes to the telomere and regulates the DNA damage response at that end. So what we're talking about here is this other molecule, BCLAF1 and KU70 and RAD50. These are all the players that are responsible with monitoring the DNA damage and making sure that there's an active attempt to heal. If there is an active attempt to heal, you get into these problems with carcinogenesis and failed apoptosis in essence. So I believe that science will work out the mechanism whereby activation of telomerase allows the more efficient workings of these existing pathways. So they have to do with telomere maintenance and they have to do with DNA repair. And as a result, a follow-up study to that earlier 2007 mice study, again, Blasco published in 2011, the aging mice, remember those G3 aging mice, she gave them TA65, and this is what she found. It's proof of what Mueller was talking about when he was irradiating the fruit flies. What she found was that when you take this is a regular aging mouse without telomerase, the percentage of telomeres shorter than 8,000 kilobase pairs is high. It's like 14% or so. And the signal-free ends are high. But when you introduce one functioning copy of the TERK and let the telomerase work, that drops down. And then this is no TA65. DMSO is just control, placebo. So just by introducing the gene, you got a drop off of the signal-free and an increase in the telomere length. When you added the single molecule TA65 to these mice, look what happened. The percentage of telomere that are signal-free dropped down, way down on this histogram, and the higher concentration actually dropped down less, which is interesting. But both were statistically significant. This 1 in 10,000 chance of being random. This 5 in 100 chance of being random, and that's the p-value. And we look at the telomeres shorter than 8,000 kilobase pairs. Again, precipitous drop-off when you gave TA65. Now, how do we know this is because of the TA65 or because of telomerase? Well, good question. The way we know that is when we look at the TERK-negative mice who have no key to operate the engine. Addition of telomerase activation, TA65, made no difference, no statistical difference. So it is mediated through telomerase activity. So therein lies the key to what we are doing here, we believe. We are allowing these cells to undergo the final apoptosis. And that really is the talk. So in summary, there are 3 commercially available tests. One, SpectraCell, which is quantitative PCR, which is, in my estimation, not as reliable or useful. There is the Repeat Diagnostics out of Canada, and it's a good study for people who are science-minded. That is the same price, but again, a much more reliable measurement of the median. And then we have the life length, which is over twice as expensive, up to $1,000, but will give you a measurement of the critically shortened or damaged telomeres, which is what we need. So that's really where the disease is. That's really where the future is, and I think it's worth the money. One thing I didn't flesh out here, which is important, is that quantitative PCR also is standardized to a single copy gene. So it's the telomere to signal, or single copy, T to S. So in the case of older people with DNA damage, it presumes that you have the right number of chromosomes, like 46 chromosomes, and they're all intact with the right number of single gene copies, which, as you can see from the confetti that is our genome as we get older, is a bad assumption to make. So you just want to be able to measure the raw telomere. You don't want to have it standardized to one telomere per chromosome, because that becomes a faulty assumption as we start to get into this mess as the cells get older. Okay, great. So that's really all I want to say on the subject. Happy to answer your questions either in email, offline, or currently if you have any questions, type them in to the chat box. Abraham asks, can you use an easy, simple way to explain how the telomere be increased? Well, that's easy. That was the subject of the 2009 Nobel Prize. That's just the engine, this telomerase, which is made in every stem cell nucleus. It gloms on to the end of the chromosome, and it adds 6 base pairs at a time. Boom, boom, boom. And the normal mechanism on the lagging strand will fill in the other complementary end. So this is always on, especially active at night, and this is normal. So what normally happens in all stem cells is telomerase is active, and it is pumping out more telomere while you're sleeping. So that's why sleep is so very, very important to repair your DNA. So, and of course, sleep being one of the most noticeable and universal improvements in telomerase activation. I just got an email from a gentleman who said that he was in hibernation for 9 days. And this is very common. The first 2 weeks you can get, if you're at the right dose. He didn't notice at the lower dose. At the higher dose, he was just sleepy. But it wasn't like he was tired. It was his body, the consciousness of the body was requesting shutdown. So you do get these people who sleep a lot because the organism is trying to coordinate, repair. And so I always stress it's very important to have a doctor to talk to, because all these symptoms that you have need to be understood, explored, and maybe you need to adjust your dosing or timing. Because if you're not noticing changes in your sleep and dreaming, it may not be the proper dose. Question. Bobby Wilder asks, how about Telome Health, Elizabeth Blackburn's lab? Yeah, so I went ahead and I've spoken with Dr. Harley, who is one of the principals in that company, and also their representative. And I really haven't been unable to get any information about what they're planning, so that will be up to them. I thought there was some attempt at qPCR, but again, they've been pretty mum on what they're planning. But Cal Harley is, of course, central scientist in Geron, and he was a co-author in one of the studies of TA65 in mice, and is a close collaborator with TA Sciences, and certainly he will come up with a test that will be valuable. But as of now, they do not have a commercially available telomere measurement, I don't believe. Okay, great. Well, a lot of the people on this call have been patients of mine, if not most, and you know, there can be errors with tests. I know one gentleman had a particularly short test, and when that happens, it's like you get this unfair sentence, and you think, it's not fair. I've lived my life, healthy, and how could this be? Yes, it's possible it was an error, but it also is possible it's not. And it's probable that it's not. There are internal controls to make sure that each batch is run and is valid. So, again, it's very important not to treat lab tests. And when I dropped from 9,100 to 8,400, I did not panic, because you have to sometimes disregard lab tests, or at least follow the trend. When we were in residency, the interns in residence got in the most trouble were those who treated lab values instead of going to the bedside to talk to the patient, listen, and understand what the history was. So very important to understand the value of a clinician. You know, a lot of people are do-it-yourselfers, and I respect that. One prominent person was just recommended TA65, and they ordered, it's a famous producer, they ordered a bunch from the company, and that's great, but unless you have somebody helping you interpret, it could be fraught with issues of interpretation of, you know, I wouldn't say complication, but because it's a very safe product, but it's nice to have somebody with you helping you, and there is no additional charge to that. We enjoy helping people. So if you were to climb Mount Everest, you wouldn't just walk up the hill by yourself. You would find somebody. You wouldn't just find anybody who was sitting by the pub. You would find somebody who'd been up Mount Everest. And I think that's really what our role here is. We're experienced Sherpas, and I don't believe that this is something like potassium, that it gets into your body and it just doesn't matter when you take it or how you take it. It is not that kind of thing. It's a functional treatment, a holistic cure, and it requires some wisdom and some experience. Everybody's very interested in this topic, I see. That's great. Resveratrol interfere with TA65? I don't say any supplement interferes with TA65. I just recommend saving your money and just buying more TA65. Do I work specifically with age diagnostic labs once the initial test is complete? If you send me the results, I'll talk to them with you. And of course, if you buy TA65 from us, I'm more happy to do so. But I'm not parsimonious with my time yet. It's not under great demand. So call me up. We'll talk about it. No problem. In the first 5 years of doing this, people would ask me, should I have my telomeres measured? Doesn't it make sense? And I would be pretty discouraging over that, knowing that the data do not bear out a predictable increase in everyone's mean telomere length. And again, there could be many reasons for that. You know, you just don't know. Like, it could be like voting in West Palm Beach. You know, if you start rescuing the older cells and old people turn up to the vote, it could skew the voting to be shorter in the telomere length. You just don't know what is going on, so you need to treat the trend and examine the trend. So yeah, don't shy away from doing the testing. I would shy away from jumping to conclusions based on the testing. I mean, a gentleman, a friend of mine I played golf with, 50-year-old CEO, very stressful lifestyle, had those shortest telomeres we've ever measured, 2,900. I mean, perhaps this is a gentleman that would have, you know, had a serious health problem soon, and he refuses to measure it again. And this whole "I'd rather not know" thinking is probably not good. In my experience in clinical medicine, those that are afraid of doctors eventually find that they've given them a reason to be afraid, given themselves a reason. They put stuff off, and then pretty soon it's incurable, whatever they were worried about. So definitely it's good to get your telomeres tested, and if it's not a great number, it's nice to follow it. And you don't just have to take T65. You can start in meditation, as Jeff says. Huge. Get off the sleep meds. Exercise. Watch your weight. Start helping other people, community volunteering. And all the good karma stuff will turn up your telomerase and make you healthier and live longer. But if you can afford it and if you believe it, TA65 can give you a little leg up too and help those little micro machines do what they're trying to do, both in terms of lengthening and perhaps even destroying damaged cells and allowing them to be replaced. All right, everybody. Hey, thanks. Have a great Wednesday. Enjoy your day, and I really appreciate it. This is Dr. Ed Park from Recharge Biomedical, wishing your chromosomes a healthy, happy future, unlike this poor, mangy dog. All right, everybody. Have a good day. Bye-bye.
All that you touch and all that you see, all that you taste, all you feel, and all that you love, and all that you hate, all you distrust, all you save, and all that you give, and all that you deal, all that you buy, beg, borrow, or steal, and all you create, and all you destroy, eat, and everyone you meet, and all that you slight, and everyone you fight, and all that is now.